This project focuses on designing and validating an expression vector for human insulin production in E. coli. Insulin is essential for managing diabetes, a condition affecting millions globally. Traditional methods like animal-derived insulin face challenges such as limited supply and high costs. Recombinant DNA technology provides an efficient, scalable, and cost-effective solution. By leveraging E. coli as a host, this approach not only enables large-scale insulin production, but also serves as a model for producing other therapeutic proteins. It showcases the transformative power of biotechnology in addressing global healthcare needs. Let's explore the details for this project. The plasmid I selected for this project is the PET28A+ vector, which is widely used for high-level protein expression in E. coli. First, it contains a T7 promoters, which enables strong and regulated protein expression. Secondly, the vector has a kanamycin-resistant gene, which allows us to select only the bacteria that successfully take up the plasmid. Additionally, the presence of a multiple cloning site, MCS, facilitates the insertion of our insulin gene, making the cloning process straightforward. The PET28A+ vector also offers some unique features such as HISTAG, which simplifies protein purification and restriction enzyme sites like as EcoR1 and XHO1, which are used to insert the insulin gene precisely. On the right side of this slide, you can see the plasmid map highlighting its key components and the layout of its functional regions. Let's begin with the primers. The forward primers includes an EcoR1 restriction site and the start codon ATG to ensure proper gene expression. The reverse primer incorporates XHO1 site and the stop codon TAA for correct translation termination. These restriction sites allow seamless insertion into the multiple cloning site MCS of the vector. The cDNA sequence shown here was retrieved from NCBI. This sequence is free of introns, making it ideal for expression in bacterial systems. On the right is the 6-step workflow for gene cloning. Number 1. Extract insulin cDNA, retrieve the intron-free insulin gene from NCBI. Number 2. Design primers. Add EcoR1 start codon and XHO1 stop codon sites to forward and reverse primer. Number 3. PCR amplification. Generate multiple copies of the insulin cDNA. Number 4. Digest with enzymes. Cut plasmid and insulin gene using EcoR1 and XHO1 for seamless insertion. Number 5. Ligation. Insert insulin gene into the plasmid with ligase enzymes. Number 6. Transform into E. coli. Introduce recombinant plasmid into component cell for expression. The ribosome binding site RBS ensure efficient translation of the insulin mRNA, playing a pivotal role in protein expression. Slide 5. Final vector map. This slide presents the final vector map of PET28A+ with the human insulin gene integrated. The multiple cloning site, MCS, INS gene location, and kanamycin resistance work together to provide a stable and efficient system for producing recombinant insulin in E. coli. Vector transfer to E. coli. We introduce the recombinant plasmid into component E. coli BL21 cells using the heat shock methods. These methods temporarily increase membrane permeability, facilitating the uptake of plasmid into bacterial cells. Colony selection and screening. Kanamycin resistance. We plate the transformed cells onto agar containing kanamycin. Only cells that successfully incorporate the plasmid survive, ensuring we work with the correct colonies. Sanger sequencing. Using extracted DNA, we validate the sequence accuracy of the insulin gene. This step ensures there are no errors introduced during cloning. Validation of expression. Finally, the production of insulin is confirmed through a Western blot analysis, which detects the presence of the insulin protein using specific antibodies. This critical step not only verifies expression, but also ensures the protein is synthesized correctly. Conclusion. This project highlights the power of recombinant DNA technology in addressing challenges like insulin production. By leveraging advanced tools and methods, we successfully designed and validated an expression vector for efficient insulin production in E. coli.